Now, if you got a 6 marker like this on enzymes, would you be able to get all the marks? Explain how the structure of enzyme molecules is related to the effect of pH on the activity of amylase. A lot going on here. So it's asking all about the structure of enzymes. So you basically need to explain how enzymes work. So this is basically explaining the lock and key theory. So how does that work? So we've got two things here. Inside the enzyme, you have an active site. The active site has a specific shape, and whatever it is that's being broken down is known as the substrate. And what happens is the shape of the active site is complementary to the shape of the substrate, so it perfectly fits in. So you can see here in this diagram, the substrate will perfectly fit into that enzyme. It's like a lock and a key. The substrate is the key, the enzyme is the lock. This substrate will only fit into this type of enzyme. So we just need to explain this to get a few of the marks, not all of the marks. So you can say enzymes have an active site with a specific shape, and that shape matches, or is complementary, the shape of the substrate. So in this case, what's the substrate that's being used? Maybe you can apply it to what they're asking here. They said, how is it related to the effect of pH and the activity of amylase? What does amylase break down? Starch. So basically this substrate is starch. You can actually mention that in the question here. So you can say the substrate is starch for the enzyme amylase. Okay, so we've talked about the structure. Now we need to talk about the effect of pH, which is asked here, on the activity of the amylase. So what happens? So there is an optimum pH where all of these reactions happen the best, but if you increase or decrease your pH around that optimum pH, so if it becomes too low or becomes too high, what happens is this active site changes shape. If the active site changes shape, it means this substrate is no longer going to be able to fit into it. That means the substrate is not going to be broken down, so your reaction is going to be much slower. So we need to apply this to amylase. So let's write this down. If the pH becomes much higher or lower than the optimum, the enzyme will denature. So that's what we call it when the enzyme's active site changes shape. The enzyme will denature and the active site will change shape, so the substrate will not fit in it. What does that mean? That means amylase can no longer break down the starch. So the rate of digestion, remember all of this is the process of digestion, breaking down starch, that's going to slow down. So we can say amylase will not break down starch, so the rate of digestion decreases. And that's how you get 6 marks there.